Hey guys, I wanted to talk about just a couple of things from chapter 27 before it got too far away from us. So, chapter 27 is about prokaryotes and archaea um, and bacteria, and it's actually not super new content, so a lot of it will be very familiar to you from previous chapters, previous courses. The only really new thing that's on the objectives that I want to make sure I go over somehow is the stuff about genetic recombination. So specifically, transformation, transduction, and conjugation. So let's do this little section as a presentation, shall we? Okay, so bacteria and archaea don't have sex in the same way that eukaryotes do. Which is to say there aren't gametes formed that fuse during fertilization. There's no meiosis, right? So this raises a really interesting question slash evolutionary puzzle, which is how do you get new genetic diversity, right? If there's a bad mutation in your mom, how do you, as a bacterium, ever escape that bad mutation, right? Are you just like doomed and all of your children doomed for eternity to have this bad mutation? And if so, why are there still bacteria and archaea given the inevitability of bad mutations accumulating over time? Okay? And the answer is because of genetic recombination. So despite there not being sex in the way that we know it, there are methods of gene exchange among archaea and bacteria. And specifically, there are three that we're going to talk about in the chapter. So all of these facilitate some kind of genetic recombination. And they are transformation, transduction, and conjugation. And all of those which cause genes to move from one lineage to another is called horizontal gene transfer among bacteria and archaea. Okay. So transformation you're actually already familiar with back from the Griffith experiment where they took the R bacteria and the S bacteria and they heat killed one and allowed the other one to be exposed to the heat killed bacteria and they took on their DNA. Okay, so that's called transformation, where a prokaryotic cell essentially takes up or incorporates foreign DNA particles into its own genome, starts expressing those genes. Okay, so that does happen. We've already talked about that. Transduction is new. So transduction is where you move genes between bacterial lineages via bacteriophages, which are viruses that specialize in infecting bacteria. So here's how that might work. Okay, you've got a phage, so a virus, that infects a bacterial donor cell. Okay, that donor cell makes two alleles, one at the A locus, so it's the A plus allele, and one at the B locus, the B plus allele. Okay? So that phage replicates inside the host, okay? Makes new viral capsid proteins, etc. And then sometimes the genome or parts of the genome of the host bacterium get taken up into those viral capsids. So fragments of DNA, say bearing the A plus allele, get packaged into the phage. When that phage goes on to infect new hosts, let's say a recipient cell, whose alleles are A minus and B minus, okay, the new the foreign DNA introduced by the virus can actually recombine with the host genome. Okay, so it's a homologous region, and you actually get recombination. So you get crossover events between this just like straggling DNA and the host circular genome. Okay, if the virus were to just like release this DNA, this naked DNA, into the host cell would eventually degrade that DNA, right? But if there's recombination, that becomes part of its genome and its children's genome, etc., etc. Okay, so incorporating that phage DNA can create recombinant cells. Okay, in this case, we have an individual now who's A plus, got the A plus allele from the donor, and is still B minus. Okay. So I hope that makes transduction make a little more sense. Another way you can get gene exchange or horizontal gene transfer between bacterial lineages is called conjugation. So conjugation is where genetic material is transferred. Okay, and bacteria, this transfer is unidirectional. So there's always a recipient cell and a donor cell. Okay, the donor cell attaches itself to the recipient by a structure called a pilus, and that pilus is a grappling hook slash also DNA delivery tube. Okay, so the DNA that it gets delivered is called an F factor for frequency. I think that's what the F stands for. Okay, and an F factor is actually required to make a pilus, which is pili in plural. So here's what a pilus looks like. So it's called a sex pilus because it's facilitating gene exchange, even though it's not technically like formal sex. This is bacterial sex. I always thought sex pilus was a great band name. Okay. So the F factor that allows a bacterium to make a sex pilus, which is just a DNA bridge, a tube slash grappling hook, okay, can occur on a plasmid, right? And if you look in the chapter, right, it talks a lot about plasmids. So plasmids are just little segments of DNA that aren't the main genome of the bacterium, okay, but they are intact, right, and they do get transcribed and translated. So it's just basically like little bonus material inside the bacterium. Okay, so the F factor can occur on a plasmid, and if the plasmid has an F factor, it's called an F plasmid. And any bacterium that doesn't have an F factor can receive DNA during conjugation. And F factors get transferred or can get transferred during conjugation. So let's say we've got a donor cell with an F factor here, and a recipient cell that's F minus, so it doesn't have an F factor plasmid. Okay, so the donor forms a sex pilus and it attaches to the recipient, reels the recipient in, and starts copying DNA into the host cell. Okay, so there is an intact copy of the F plasmid that remains in the donor cell and a copy that ends up in the recipient cell. Okay, so basically the recipient cell synthesizes one complementary strand, and the host cell, the donor, synthesizes another copy. So they're both synthesizing a complementary strand. The donor is giving one of the strands to the recipient. At the end of this conjugation event, now both cells are F plus and can both go on to form sex pili with F negative cells nearby. Okay, occasionally the F factor actually can end up in the main genome, in the circular chromosome that is the bacterial genome. Okay? And in this case, you can actually get transfer not just of F factors, but lots of genome content from donor to recipient. Okay, if a bacterium has the F factor in its genome, it's called an HFR plus or an HFR cell, HFR being high frequency of recombination. So the donor will form a sex pilus, reel the recipient in, okay, and will start moving one strand of its genome through that sex pilus. Okay, at some point, because remember, this is a pretty large molecule, right? The entire genome of a bacterium, that's a lot of DNA. Even though it's bacterial, right? That's still a big molecule. And usually what happens is this conjugation event is interrupted before the transfer of the entire genome is completed, right? So usually the donor will only donate part of its genome, including the F vector, into the host cell. Okay, so this is naked DNA, right? It can't really circularize as easily as just a plain F vector could. So it's relying once again on recombination events, just like back in transduction. So there are recombination events, so there's crossover events between the host's the recipient's original genome, and now part of the donor's genome. So if the host was A minus, had the A minus allele, now maybe after a recombination event it could have the A plus allele. And enzymes degrade the non-circular linear DNA that doesn't get incorporated into the genome. Okay, so this stuff just gets chewed away. But now we've actually transferred an A plus allele to the recipient bacterium. Any plasmid that carries an antibiotic resistance gene is called an R plasmid among bacteria, right? And remember, antibiotics kill sensitive bacteria, but not bacteria that have R plasmid specifically for those particular antibiotics. So over time, in selective pressures where antibiotics are being used, the frequency of individuals bearing resistance alleles increases. And part of the reason it increases is because bacteria are exchanging these R plasmids via transduction and conjugation and transformation. Okay. So if you were ever asked a question about like how horizontal gene transfer or how genetic exchange works in bacteria, there are 3 modes that your book talks about: transformation, transduction, and conjugation. All right, and I think that's the only truly very new stuff from this chapter. Thanks a lot. Take care. Have a good day.